Welcome back, ladies and gentlemen. Today we're going to be taking a look at hydrocarbon isomers. Now, you may recall an isomer is the organic chemist way of saying an allotrope. They're different, but they're very, very similar. So an allotrope, as we learned in physical properties of matter, is where you take the same building blocks and you assemble them in different ways. So if I take carbon and I lay it out in big sheets, I get graphite. Well, I get graphene, and I can stack them to make graphite. If I take them instead and arrange them in a tetrahedron, it's a type of shape, I get diamond. Both of these behave very differently. Graphite is soft enough you can write with, diamond is hard enough to cut through anything. But they're both made of carbon. Now, we have carbon in our organic compounds, but we're going to be taking that a little bit further. I'm not going to be just working with carbon here, I'm going to be working with hydrocarbons, carbons and hydrogens connected together. So I take these hydrocarbons and I assemble them in pieces. What's an isomer? It's when I take a group of carbon-hydrogen, right, a hydrocarbon piece, and I attach it in different ways. Do I put them end to end? Do I put them some this way, some this way? How do I do it? So instead of working with individual carbons, I'm working with groups of carbons plus hydrogens. And that's what an isomer is. It's still basically the same as an allotrope, but it's got more pieces involved. Let's take a look at some examples, okay? Here's one. Previously, we discussed two isomers of C4H10. That's a bit of a lie. If you check the previous video, we did C5H12, but shh, don't let anyone know. Here we've got butane, right here, very simple, right? We've got 1, 2, 3, 4 different carbons. Over here, we don't have that. Over here, we've got 1, 2, 3, 4, one of them sticking out down here. So over here, each of these two central carbon atoms are mostly attached to two other carbons, but mostly to hydrogens, whereas this guy here is attached to 3 carbons. It's very different behavior. So these are the starting point of isomers, and they're pretty basic, but we can go deeper. We can go more complicated, which, I mean, it's no surprise to you guys, right? I mean, every time we've dealt with this stuff, it's always gotten more and more and more complicated, and yet the names are always just as bad. Chemists, they gotta get a poet in here. Okay, let's take a look here. Isomer conformational. There's a big word. What does that mean? All particles are in constant motion. We know this from the kinetic molecular theory. All particles are in constant motion, including those that are part of molecules. So much as I am moving around up here as part of the video, much as you would move around in your desk as part of the crystal lattice of the classroom, as we talked about in physical properties, particles in a molecule, be it organic or inorganic, will also move. What would that mean? Well, carbon atoms can rotate and shift about. They can wiggle, basically. And that this rotational motion results in different conformations, or shapes. It's a big fancy word to say shapes, but are actually all identical in terms of how they behave chemically. What do I mean by this? Okay, conformations convert rapidly from one to another and are just different perspective of the same structural formula. So it's kind of like I'm saying, behold, this is one of the conformations of the marker clicker structure. Just work with me. This is one of the conformations. Now I will make a new one. Ooh, look, it is now so small in your screen you can barely see it. And now it is big again. All I did was turn it, obviously. But when the particles are all jiggling around, moving like crazy, sometimes they're going to look in different arrangements. Sometimes they're going to look like this, sometimes like this, sometimes like this. And they all kind of look different. And this is pretty rigid, right? I can only really rotate it as a group. But what happens if, like, this piece could rotate or spin or move in some way? It's going to look different, but it doesn't actually behave any differently. It all sort of behaves the same. Now, this is an example. These are all butane. Okay, over there, right on the far side, here we go. We got our just a straight line, 4 in a row, boom, boom, boom. But I could have like a zigzag, like this. Or I could have a boom, 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 boom. This is like a chair kind of shape, or like a half square. And then you got all of these. Look at these. These are mostly straight, but then this part sticks out a little bit. Well, what's the difference between that and this, right? Like if I take this one here, the second one, if I take this one here, and I just take that and like rotated it, I could make it fit right into this one here. Right? I could move them and put them over top of each other. Being able to put things over top of each other, we call that a superposition. Like you can just superimpose them on top of each other. I can't do that with my hands. Like I can do this, but I can't have this because my thumbs are on the wrong side. We'll talk about that in a little bit. But all of these conformations, all of these shapes are identical, even though they look different. They behave the same, though. Right? This can make it tricky to identify organic compounds. Now, to be fair, we saw this already when we were trying to identify them in the naming stuff, right? Sometimes we'd have to just like read straight across, but sometimes we had to do these weird zigzags in order to find the longest chain. And it's like, Mr. C, why aren't they just straight lines? Nature doesn't work that way. Things wiggle. Things move. I mean, students won't sit still in their desks. Why are you surprised that chemistry won't sit still for you? Sorry, it's just how it is. But on the plus side, at least we have the tools necessary to start analyzing this stuff. That makes things a little easier. These are not conformational isomers of each other. Why? Okay, this is a straight line written in the condensed form. This is a straight line in the condensed form, but instead of having the last one here on the end, it got pulled off and put here. There is no way for me to rotate this molecule here and make this one. In order to make this one, I would have to pluck this piece off and move it over here and reattach it. That requires a completely different shift of everything. Same with this guy over here. I've taken another one off the end, right? I've grabbed this one. Oh, can I get my, there we go. I grab this one and I just take it and I stick it here. Boom. Oh, wait, would that work? No. What? Why? What? How? Let's see. Need the markers. Should have grabbed mine. Oh, well, let's try this one. If I take this piece off here and I stick it here, well, what does that do? Let's try drawing it. We're going to have 1, 2, 3. 1, 2, 3. This one here is sticking up like this. Now, I'm going to pluck off the other one and I'm going to use red. So the red was here. I'm going to pluck it off and put it here. I mean, that looks different, doesn't it? But if I circle the longest chain, that one down there is just part of the longest chain, right? I can just be like, oh, okay, well, there's the longest chain. And this is just one sticking out the end. Well, that's the same as this. Oh, I didn't actually make anything different. So what else would I have to do? Well, look at how I went from here to here. I didn't just pluck one off. I plucked both off. And then I stick this one over here to make this T shape, the plus sign. So these conformations can be tricky. Figuring out what is actually a different material, it's not easy. You've got to really look at it to really get it. You got to understand. And you got to tinker with it. You got to get your hands dirty, mess around, move things around, redraw it, re-sketch it out, remodel it. Yeah, there are kits for this. They're called modeling kits or molecular modeling kits. Their purpose is to help make it so we can see these things because, you know, barring the occasional person in my class who's actually a hidden superhero and who can shrink down to the size of an atom, we can't see what this stuff looks like. We have to infer based on science, knowledge. So, isomers, structural isomers. If you cannot rotate the carbon to get the same structure, then you have a structural isomer. This is different from a conformational isomer. Conformation means shape, right? If I can slightly adjust the shape, right, tweak a bit so it's like instead of it being a straight line, it's bent at the end, it looks different, but it's not really. These ones here, structural isomers, are physically different. So for the purposes of a conformational, a shape isomer, it could be something like shoes, right? You, your friend, they both have shoes. They both do shoe things. They look different, but they're still both shoes. A structural isomer is shoes and gloves. They both put the 5 little digits inside of something to keep them safe, but one of them works on hands and one of them works on feet. So they're different. It's not the best analogy, but it's a start. So conformational isomers, I can rotate to make something new again. Structural isomers, I have to pull apart and reattach. That's the big difference, okay? Structural, you have to dismantle. Conformational, you have to rotate or twist in some way. Structural isomers are compounds that have the same molecular formula, but different structural arrangements. Oh, see, I got C5H12 again. I must have changed my notes at some point. Structural isomers have different properties from one another. This helps to distinguish one isomer from another, as they would both have the same molecular mass. So if I wanted to try to separate these guys based on how much they weigh, it wouldn't work because they weigh the same. But some of them will behave differently. We've already talked about this a little bit in class, and we talked about the fact that isomers that are very flat, for example, have an easy time stacking on top of each other. Right? They're like sheets of paper. I just lay them one on top of another. Structures that have got bits that stick out, well, when I try to lay them on top of the other, they're not going to fit. Right? They're going to get jammed in there. They're going to rub against each other. Stuff's going to be sticking up. They're not going to sit flat. So that's going to be a big difference, the way that they behave, the way that they interact. But let's take a look at some more examples here, okay? The greater the number of carbons in the alkane, the greater number of possible isomers. 4, you can have 2 isomers. 5, you can have 3. 6, you can have 5. It's not an exponential curve. It's more of a geometric curve, I think. It's linear, at least. Not geometric, I think. So some drawing tips, okay? Start by drawing a straight chain hydrocarbon. Right, so if a question says, draw all of the isomers, the structural isomers of a particular pentane, sure, whatever, just choosing one at random. Draw all the structural isomers of pentane. How would I do it? Okay, step 1, draw pentane. Brilliant. I'm going to start with the first one. Step 2, attach an alkyl group to each of the central carbons one at a time, making sure that each new structure is not a conformation, a rotation, of another. Step 3, attach 2 alkyl groups to each of the central carbons one at a time. Attach 2 alkyl groups to 2 different central carbon atoms at a time, etc., etc. You noticing a pattern here? Okay. Normally I would pull out the modeling kits and let you guys try this now. Let you guys make all the isomers of heptane. I still would encourage you to try drawing it. To sit here, okay, heptane, that's 7. Okay, so we get out, where's the marker? So we're 7. So 1, 2, 3, 4, 5, 6, 7. This is one of the possible isomers. Now, what did we say? Okay, once you've drawn out 7, your next step is that we take 1 off the end and we put it in one of the middle ones. Okay, so I'm going to take it off of the end one here. So I'm going to go, okay, so 1, 2, 3, 4, 5, 6. Okay, because I'm taking 1 off, I take it off the end, and I got to put it in the middle. Not this one. This is the new ending. So I got to put it somewhere in the middle. Not here. That's just the end again. The middle ones are these 4 in the middle here. So I'm going to do 1 there. Right? So I test each one after another. Oh, let's make this a little darker so we can see. Okay? So we attach it here in the middle. That looks new, right? Like I can't rotate this to make the first one again. Okay. What happens if I put it somewhere else? Okay, wait, here we go. 1, 2, 3, 4, 5, 6. And I'm going to take this one off, and I'm going to put it somewhere else. Okay? Now, I could put it anywhere, right? Middle one, got to be middle. If I put it on the end, I just make the first guy again. So I could put him on the middle, and I could put him the next one. That'd probably be a good way to do it because it keeps things consistent, right? I do the next one, then the next one, the next one. What happens if I'm a little bit silly and I jump all the way over to this guy? What did I just do? Did I make something new? No, right? Because I can take this second one down here, third, I guess. I can take this one and flip it like this, and it would be this guy. I could remake it. Well, that's not helpful. I just made the same thing twice. So do it in order. Right? What did the rules say? Make the chain, take one of the ends off, put it in the middle. Then move it to the next middle. Then the next middle. See if it's different. Once you've done that, take two off. Put them on the same one. Move them both, move them to the same one again. Do the same one again, check if they're all different. Then, two off, put them on two different ones. Then two different ones again, and two different ones again. Right? Check if they're all different. Test it one after another, after another, after another. You can try taking 3 off, 4, 5, 6. Of course, the more you take off, the more likely that what's going to happen is you're just going to make a new, really long chain that's just going to be zigzaggy. But all right, give it a try. I want you guys to attempt this. I know we don't have the modeling kits. It's not as exciting. But what do we get? Well, here's the answers. Are we ready? Have you done it? Have you tried it? I hope so, because here we go. There they are. 7 in a row. 6 in a row with one sticking out. We did that one. 6 in a row with one sticking out, but not on the second one, on the third one instead. Then I have 5 in a row, 2 sticking out. 5 in a row, 2 sticking out in different places. 5 in a row with 2 sticking out on the same one. 5 in a row with 2 sticking out on the same one in the middle. 5 in a row with 2 sticking out all together. It's an ethyl instead of a methyl. And then finally, you got 4 in a row with 3 total sticking out. There's a lot of options, right? See if you can see it better if I'm standing here. There's a lot of options. It's because it's a pretty big molecule. Now, again, each one of these is going to have a different behavior, right? These ones up here are going to stack together really well. These ones over here are going to stack together terribly, right? If I want to stack these guys up, I have to make sure they fit together just perfectly. It's going to be like trying to fit together, trying to think of an example, hockey sticks, right? You got a bunch of hockey sticks, you're trying to put them away in the bin. Well, if some of them are pointing down, some of them are pointing up, the ends are going to get jammed together. They're just not going to fit. I mean, that's not a great example. That'd be a better example for like this guy. But whatever, we did it. It's going to be hard to get them in there. So you're going to have to be careful, and that's going to change how they behave. Okay, all right, we saw this. So we've had conformational isomers, same formula, rotated structure. Structural isomers, same formula, different structure. Okay. Now we have stereoisomers. I swear it's like the chemists are trying to make us pull our hair out. Maybe that's why I lost all of mine. Isomer. Okay, let's break it down. We've got the word isomer in here. That's at the end. We've also got the word stereo. What does stereo mean? You got stereo headphones. I don't have stereo headphones right now. I've got a mono headphone. Can you see it? It's right there. That's what you guys are getting the sound through. Mono headphone. We know what mono means. Stereo means 2. Stereoisomers are molecules where the atoms are joined in the same order, but the positions of the atoms in space are different. What? What does that have to do with 2? This is what it has to do. These are called geometric or optical. Optical. Okay, let's break them down. Geometric isomers are when a double bond is present in a molecule, and the carbons then lose their ability to rotate. Basically, if they've got 2 hooks on each other, right? They got 2 hooks connecting, they can't spin. They don't have any room. If there's only 1 hook, they can spin a little bit, right? Therefore, groups on either side of the double bond can have a different orientation in space. Let's just show you a picture, okay? For example, Both the following are 2-butene. Butene, E-N-E, butene, double bond. 2, the double bond is on the second one. Both of these are 2-butene, but they look different. Right? This one up here, the two are going down together. This one over here, they're going one up, one down. Right? It's like this versus like this. The problem is I can rotate my arm because I don't have a double bond connecting these two. If there's a double bond, they'd be stuck. I couldn't rotate it. It wouldn't work. Now I'm doing the robot. So what's happening here? The double bond stops them from being able to rotate, which means that they get locked in whatever position they were. I can take the entire molecule and rotate it, right? Like this one here is down. I could flip the whole thing up, but I can't flip one up and one down. Doesn't work because I can't rotate around that bond. This is critical. Actually, this is really critical. We need this to live. Wait, what? Trans, cis, where have you heard those words before? A couple different places. Probably biology, probably family studies, but they actually come from science. They're Greek again, because the Greeks, of course. They use prefixes to denote same or opposite, and they do it with Greek. Cis means same. Trans means opposite. Now those other terms you've heard make a lot more sense, don't they? This is another way to draw that. So if we look here, right, cis, they're on the same side. Trans, they're on opposite sides. Cis is the same, trans means opposite. So trans fat just means opposite fat. Fat that is like this in some manner. Not this, but this. Could be this instead. They're opposite. Hands are on the opposite sides of my body. One's up, one's down. The reason that trans fats are bad has to do with biochemistry. Bio meaning living, chemistry meaning study of matter. Biochemistry is, of course, the study of chemistry in living organisms. All fats made by organisms are cis. All of them. Period. Done. Every one of them. All of them look either like this or like this. But we as humans are very clever. We have the ability with chemistry to make trans fats that are this way. Our digestive system isn't designed to process trans fats so they don't move. Trans fats, once they get in your body, cannot leave because there is no system in your body that can pick them up. We have hands, right? We can just pick stuff up. But if I ask you to go pick up water with your hands, you're going to have a hard time, right? You can sort of scoop a little bit in the palm of your hand, but you can't, like a glass is going to work better. You can't pick up the water. It runs through your fingers. Our hands are not designed to pick up liquids. They're designed to pick up solids. The parts of our body are designed to pick up cis fats, fats that are like this or like this. When humans, very clever humans, make trans fats, those trans fats are like trying to pick up water, except even a little worse because we can't do it at all. So the analogy here, as we see it, is that when we try to pick up trans fats, they just fall through our fingers. We can't do it. But that's bad because you eat them. You put them in your mouth, you put them in your stomach. Where are they going to go? What can they do? They're stuck. They can't go anywhere. Trans fats end up getting inside your body, and once they're there, they're stuck there. They cannot leave. And that is problematic because they gum up the works. They get in the way. They cause trouble. So here are a pair. Can we name them? Do we know how to do that? Okay, well, we got the words cis and trans to throw out there. So let's give it a shot. Okay, first things first, right? Can we do this easy? This is on the same side. That's on the opposite side. So we know this one down here is going to be cis. And this one over here is going to be trans. Okay? Now we just got to figure out how to put the names together. Okay, well, we know how we name things generally. We name them based on the number of carbons. So let's count. We've got here 1, 2, 3, 4, 5, 6 carbons. So this is going to be hexane. Well, it would be hexane, but there's a double bond right here. This is a double bond, so this should be hexene. Where is the double bond? It's not the first one, it's not the second one, it's the third one. And it's, of course, cis. Cis 3-hexene. I think that's right. Let's check. Hey, look, it is. Cis 3-hexene. Over there, we've got the other one. It's trans, as we said, and the bond is still on the third possible bond position, but now it is heptene because there are 7 carbons. Okay, so this really didn't change much, really. Like, we already did that part. Well, we already did that. We've just added one extra bit where we basically describe the stuff around the double or triple bond. Is it on the same side or opposite sides? I'm going to make a dance routine here. Boop, boop, boop. That's all we're looking for. That's not really so bad. It's kind of weird they called them geometric isomers. Just wanted to use big words, I guess. All right, optical. Okay, optical, it's like optometrist. Means sight. So what are we talking about here? Optical isomers. When a carbon atom has 4 different atoms or groups attached, it is called an asymmetric or chiral carbon. Chiral. That's how you pronounce that word. Chiral. It's weird. You'd think it'd be churl, but it's chiral. It's Greek again, I think. It is similar to your right and left hands. Even though they contain the same parts, you cannot superimpose them. I can't put them over top of each other. What I mean by that is I can do this, right? Thumbs are in alignment, fingers are all in alignment, palms are together. But the 2 insides of my hands are facing each other. I cannot put it so the back of my hands are facing the same way and have all my fingers line up, right? My thumbs are on opposite sides now. Let's see, what's another example? Here we go. 2 markers. I can stack them like this, right? I can stack them where the bottom and the top, bottom, top, bottom, top, bottom, top, bottom, top. I can't do that with my hands. If I do that, they fall down. If I do that, they don't line up, right? My hands cannot cover each other's thumbs. I can do it this way, but doing it this way would be akin to me taking the markers and stacking them like this, which would be cool if I could get the balance. No. It'd be stacking them like this, which is sort of stacked, but it's not the same. So chiral molecules, or optical isomers, are kind of important because, once again, biochemistry. Oh yeah, both the structures in this picture have the same overall pieces, but you can't make one into the other. There's no way to do it except by taking a reflection, just like your hands, right? If I put my hand in a mirror, I'll see the other hand on the other side of the mirror. Now, this is important for organic chemistry, for biochemistry, for the same reason that cis and trans fats are important, right? Our bodies cannot process trans fats. Can't do it. Our bodies are designed to work with left hand. Yes, we use the hands to indicate direction. Our bodies are designed to work with left hand sugars. So sugars that have got their bits oriented in this form, not this form. So I got that right? Yeah, this form. This is left. I know left. Don't look at me like that. What that means is that if you were to eat a right-handed sugar, you can't process it. It will not interact with your body. It's the same again as us trying to collect water with our hands. Our biochemistry is not designed to handle that, so it cannot interact with it. Human life is very specific. Cis fats, left-hand sugars, nothing else. I think this is actually a plot point in Mass Effect. Whatever. Point is, these are the 3 different kinds of isomers, okay? We've got the conformational isomers that are not really different. They just look different, but you can rotate one of them into the other because the bonds will freely spin. You've got structural isomers, where you pluck pieces off and reattach them in different places. You've also got the stereoisomers, where you can have the geometry, where you can either be this, or this, or this, or this. Same side, different sides. And you got the optical isomers, which is basically the same as the stereoisomers, except that they're like on a smaller scale, right? So the stereoisomers are like all these different, this big long chain of carbon going down or going up. The optical isomers are where I've got the single carbon in the center, and do I put the pieces on in this order, 1, 2, 3, or do I put them on 2, 1, 3? And then it changes the order a bit in terms of when you're looking at them. And you can't make one into the other, just like you can't make one hand into the other. Okay, it's a lot of words, a ton of words, but they're words that are important. They are all based on certain aspects of our biology. We need them. Things like the trans fat, for example, that's a famous one. We figured out how to make trans fat. Well, no, you know what? I've said enough already. And we'll get to how we do trans fat later on when we talk about the next topic, hydrocarbon reactions. So I'm going to leave it there, and I'm going to see you guys next time.